I'd like to begin my talk by explaining these two pictures that I've chosen for my opening slide. The first picture is of me during my PhD at Notre Dame. This is an American chestnut, one of the only surviving adult American chestnuts in northern Indiana. This was once a dominant tree species throughout the eastern forest, but a fungal pathogen that appeared in the early 20th century has almost eliminated this species from its natural range. This other picture was while I was in France. This is Eryngium alpinum, also known as the Queen of the Alps. It's a perennial species adapted to these high-altitude meadows. These meadows are maintained by either grazing or occasional, they're created by occasional avalanche falls. And when that's not happening, the forest comes in and encroaches on the habitat, eliminating this local population, which new populations are created by more avalanches. So we have a sort of classical metapopulation system for this species. Now I'm interested in the genetic consequences of these kinds of ecological change. So recent population declines, population colonization. So that's the focus of my empirical work. I'm moving into, like Paul said, some issues of when we're doing this empirical work or when we're interested in conservation collections, what is the most effective and efficient sampling strategy to use? My talk is in 4 parts. I'm going to cover about 15 minutes on the background to conservation genetics issues and some of my work as a PhD student. I'm going to then talk about my time in Europe when I worked on optimizing sampling strategy when you're doing a genetic study. And then the work that I have proposed to do at NIMBioS, collections for seed conservation, and about 10 or 15 minutes of the results that I've gotten so far. I use the phrase, and a lot of people use the phrase, genetic resources, for a couple of reasons. Genetic diversity, as we've known for a long time, is a reservoir upon which a species can draw in order to adapt to ongoing ecological changes. There's an increasing body of evidence that shows that genetic diversity contributes to various ecosystem processes and ecosystem resilience, the ability of an ecosystem to withstand or to bounce back from disturbances. We also know that genetic diversity may contribute valuable traits to agriculture, such as drought resistance. And then we also know that small populations that are lacking in genetic diversity may experience some detrimental effects of inbreeding. So these are some of the ecological and evolutionary reasons why genetic diversity is important. There's also increasingly a number of legal and policy frameworks centered around preserving genetic resources, including the U.S. Endangered Species Act, the Canadian Species at Risk Act, the International Convention on Biological Diversity, which has been signed by 192 nations. The U.S. is not among those, but the CBD recognizes that genes, species, and ecosystems are the 3 fundamental levels of biodiversity. The United Nations Food and Agriculture Organization is also interested in monitoring how these genetic resources might change over time. So if you want to know a little more about that in terms of the policy background, this is a paper that myself and my European colleagues put out last year. Importantly, one of the more recent policy drivers behind protecting and conserving genetic resources is the new targets put forth by the Convention on Biological Diversity, one of which is to hopefully be achieved by 2020, what they refer to as minimizing genetic erosion and safeguarding genetic diversity of not only agricultural related species, but also their wild relatives, and culturally or economically other economically valuable species. So given this, the ecological and evolutionary importance of genetic diversity, and given an increasing policy focus on genetic diversity, what I do as a conservation geneticist is I work to better understand, predict, and mitigate potentially negative changes in genetic diversity caused by ecological processes, such as those that are affecting the American chestnut or other species. Now, the CBD and a lot of policy documents might use vague terms like genetic erosion, but we should know that there are sort of 4 specific changes that a species' genetic composition might undergo. And those are sort of the direct loss of diversity, drift, which is an undirected change in allele frequencies due to sampling every generation. We've got directed changes that might occur through intentional or unintentional selection. And then admixture, that's hybridization between species. So that's an overview of the importance of genetic diversity. And now I'd like to introduce you to the species that I did my PhD on. This is butternut. It's an early successional riparian adapted tree species that occurs throughout the eastern U.S. forests. Tennessee is at the southern edge of the species range. The northern edge goes up into southern Quebec. The species lives for about 50 to 70 years. And what I'm going to do now is talk about 3 studies that I did during my PhD related to this species. I'm not going to go into the details of the analysis or specific results. I just want to give you some of the general conclusions that I reached as a sort of gateway to my further work that I'll spend most of the talk on. So butternuts, you can find them in the forest. You can find them in Tennessee. They exist in the Smoky Mountains, but they are increasingly rare due to this disease. You can see on this particular tree these big brown and black patches where the bark is falling off, and you've got these big open wounds. Similar to a disease that devastated the American chestnut, there's been a fungal pathogen introduced. First appeared in the southern portion of butternut's range. This is Tennessee here, and has progressed northward and is now found throughout the species range. I was interested in whether this disease had led to much loss or much change in the genetic diversity of this species. But at the same time, we have to consider this ongoing disease process in light of other processes that have also affected the range-wide diversity of this species, one of the most important being that half of the species range from about here northward has been colonized since the last glacial period. And there are other ongoing processes, such as maybe the populations that persist on the margins might be kind of smaller and more fragmented. So what I'm interested in broadly is what is the relative effect of these current and historical processes that might affect genetic diversity. And my study showed that by far the colonization northward has left the largest genetic signature across the range. Sort of the broad conclusion of that is that large shifts in a species range can leave genetic diversity impacts that last hundreds of generations. Furthermore, we have to consider current and ongoing processes in light of a species history. So that dealt with the broad range-wide genetic diversity. I'm also interested in how local environmental factors, such as the geometry of a population, the density, the clumping of individuals within a population, the habitat type, recent demographic changes, how these affect what we call local spatial genetic structure. So working with some of the same populations and some other butternut populations, I showed that there is a correlation between some measures of spatial genetic structure, which builds up through various habitat influences on seed dispersal. So there are strong correlations to the spatial arrangement and the habitat type, and some weaker correlations to recent historical changes in the population, such as colonization. And then the last study that I want to mention today is regarding hybridization between species. So butternut co-occurs in parts of its range with an introduced exotic tree species with which it can interbreed. And my work on this hybridization has showed that in locations where there's sort of the interface between farms and forests, these sort of highly modified habitats is actually where we're finding most of the hybridization, in contrast to very low rates of hybridization in continuous forests. So that was just to give you a sort of overview of the processes that I'm interested in. The processes, these are three realms that a lot of conservation geneticists are working in. For these kinds of studies to be effective, for these kinds of studies to give us correct information that can inform conservation actions, these studies need two things. That is, good analytical tools that can allow us to detect processes like migration or hybridization. So we need good analytical methods. We also need good sampling protocols that can help ensure the appropriate collection, analysis, and interpretation of genetic data, again, in order to inform conservation actions. And so what I'm going to talk about for the next maybe 15 minutes is how do we ensure that a given conservation genetic study, like those I did for my PhD, how do we ensure that these kinds of studies or experiments have enough samples to detect whatever genetic effect we're interested in? Now, some people might argue that now that we're in this sort of age of genomics, now that we have these tools such as next generation sequencing, we can sample ancient DNA to get a picture far back. And we have more and more markers that we can genotype cheaper and cheaper. So some people would say that in this time, we don't have to worry, or we have to worry less about study design. In my opinion, as we get more types of genetic data and more amounts of genetic data, we have more options for the study, and increasingly limited funds. I think in this time we actually need to be even more concerned about avoiding over or under sampling for a study. And so having an effective and efficient sampling design is of importance to several kinds of people. Maybe foremost to those who are doing the study design and executing the study, but also to the people who are writing grants. So in a grant, you present your study design and you try to argue that this will actually work. And so having evidence that your sampling strategy could be effective is important. Maybe also important to the people who are doing the evaluations of those grant applications. Unfortunately, many studies, many population genetic studies are undertaken without really much certainty that the sampling design will be successful, and certainly without knowledge of whether it would be the best sampling design. There are three ways that people go about typically doing a population genetic study, and that is to sample as much as possible, to take some guidance from recent successful studies in whatever topic you're interested in and use that as a guideline, or to follow these sort of generally accepted minimum sample sizes that the field has come to agreement upon over several years or decades of using a given technique. Now, during my PhD, I had to develop some genetic markers myself, which takes about 6 months to a year. And so before embarking on this work, I wanted to ask some advice from some professors about how many do I need to develop. And the answer that I got was to look at the journals that I want to be published in and sort of look for the average number of markers that most people are publishing with. And that's ultimately what I did. And this was sufficient for most of the work I wanted to do, but it's not really a very good answer to that question. Now, we might expect that the power of a study, the power to detect a given effect size, would be some function of the number of genetic markers used, so that's the resolution of the study, as well as a function of the characteristics of those markers, so that's number of alleles per marker, the number of samples we use, and then, of course, the strength of the signal that we're trying to detect. We might also further expect that as we increase the sampling effort, which I'm lumping several things together there, number of populations, number of individuals. So as we increase our sampling effort, we might expect an increase in the power of the study, but that relationship might take several forms. So ideally, you could create something like this before you do a study. Ideally, it would be nice to know what that relationship is between power and your sampling effort. So my main project during my first postdoc was to develop some software that would do just that, to allow you to calculate the power before you embark on a study. So this software allows a population genetic investigator to calculate the power of several study goals, including detecting a bottleneck, which is a recent drastic change in population size, successfully making assignment tests, assigning an individual to a population, using ancient DNA to detect past demographic changes in the history of a species. I'm going to explain in a sort of broad way how one can make an estimate of the power of a study. So first, you're going to simulate genetic data under a given demographic and evolutionary model. So this is our representation of the real world as it is. We're going to take a given number of genetic samples from this simulated real world and analyze those simulated data as we would in the real study. So we're going to use the same analyses that we would, or that we plan to use in the real study, and reach a conclusion based on that simulated data. Say we're trying to detect a bottleneck, this recent change in population size. That might be our conclusion based on this simulated sample. Okay? We're going to repeat that process many, many times, some hundreds or thousands of times, so that we can count the number of times that this simulated study reached a correct conclusion. That proportion can be considered an estimate of the power of the study given this model and given the number of samples we used. We then repeat this whole process for each possible sampling strategy we might be considering. So all of the different maybe numbers of markers that we think would be feasible given our resources. And then you simply compare the power of each strategy. In sort of list form, that was the graphical form. So in list form, again, we're creating a realistic model of the world as it is, genotyping samples from that synthetic system, performing a test, statistical test on those samples, repeating this loop, and calculating the proportion of those simulated studies that were successful. There's one problem. I mean, this is a really nice approach, but the problem is how do you create this simulation model in the first place? We generally don't know a lot of the parameters of the populations as they are. Okay, that's one reason why we're doing the study in the first place. So what this process, this process I just showed you, what this gives you is this conclusion. If your simulated population, if your model is accurate, so if a given change occurred as it occurred in your model, then the power that you calculate represents your ability to detect that effect, okay? So I think maybe it's obvious that what we need to do is repeat that whole simulation process over maybe a range of plausible parameter space, different demographic histories, different migration rates. If we're interested in detecting local adaptation, we would simulate different strengths of selection to basically explore some uncertainty around our power estimate. So again, at these given parameter values, our power is this amount with this sampling strategy. And so now I'll show you an example of how that works. What I'm going to do is show you a simulation. What we're going to do is simulate two demographic histories that of hypothetical species that have gone through a bottleneck similar to those of these two European species of conservation concern, the bison and then the Iberian lynx. These two species went through different demographic histories, so different decline over different number of generations, a different low population size, and then one species has made a partial recovery and the other species has not. We've got two quite different histories for these two hypothetical species. We also have some uncertainty about historical population size. So we're going to use a sort of really large number and then maybe more moderate number for the historical population size. So we'll have two models for each species. And then we're going to test a number of different marker and sample combinations. It's very apparent that the two species show very different power for this study. Furthermore, it should be apparent that so the bottom part of these two well, let me start from the beginning. X-axis is the power of these various studies. The y-axis is the different sampling strategies that we've simulated. So this is increasing the number of samples, increasing number of markers, increasing number of samples. For the two scenarios that we simulated, so assuming a smaller historical population size that's a weak bottleneck, and assuming a larger historical population size that's a strong bottleneck, what we can do is compare the power of each of these sampling strategies. We see that for the bison, under a strong bottleneck history, all of the sampling strategies have great power. Under the slightly weaker bottleneck, a different demographic history, we still see that most of them are performing quite well. On the other hand, for the lynx, we see that under the weak bottleneck, really none of these are sufficient. None of them exceed about a power of about 0.6. A couple of these might be sufficient, but for the most part, with this goal of detecting the bottleneck, for the most part, these sampling strategies would not be successful for the lynx. A couple of other things to notice, we see that some of the sampling strategies have almost equivalent or totally equivalent power, but this one might have 20 samples and this one has 40. So in that case, is it worth it to sample 20 more individuals to just get, that much increase in the power of the study? Probably not. We also see in some cases, it should be noted that as you add more samples, you might see a linear increase in the power of your study. In other cases, you don't see a linear increase. So what we've done, yeah. The power estimate, I mean, there's some randomness in the simulation itself, right? That's why we're repeating it many, many times. There's stochasticity involved in each simulation. Uh-huh. Yeah. Right. Right. Yeah. So no, this doesn't show any error bars, and so there's not we're not looking at a statistical significant difference between these things. These are estimates not for like they're estimates to be used as sort of a guide, and yeah. Right. Yeah, exactly. Exactly. So you would expect maybe a certain number of significant results just due to the stochasticity of the genetic and demographic processes. Yeah, so here what you're seeing is actually a decrease in power as you add more samples. But I mean, it's so far low that, yeah, these are just stochastic things. And the same here. Now, what we've arrived at is we've used a simulation-based approach to estimate how well a given sampling strategy would perform under a given situation. Now, this approach, this process has been used actually for several decades in population genetics, but for different purposes. So people fairly often might use the simulation-based approach to, after the fact, justify their sampling strategy to show, okay, these results really are believable. This approach is also used to test out new statistical methods to show that the method is actually precise and accurate for doing what it's supposed to do. But as yet, people are really not using this approach to actually plan a study, which would be an ideal way to go about things. So these two studies have used my software, but in the sort of more post hoc way of justifying their sampling strategy. One more study I did, one other thing I did with this software I want to mention. I'm interested in these sort of possibilities of next generation sequencing, of adding dozens or hundreds of markers to genetic studies, but it should be, we should think about when that is necessary, when it's really useful to keep adding more markers, more effort to a given study. So I did some simulations of various parameters of bottlenecks as we go from, this is the sort of normal range of a population genetic study today. This is maybe what they will be coming in the near future. So the question was, do we actually get more and more power to detect bottlenecks as we use more markers? And the answer is sort of yes. These are two different methods. This method typically outperforming this method. You can see that the stronger bottleneck typically see higher power with. And then one other thing, this dotted line. So this dotted line with the triangles and the solid line with the triangles, those are the same parameters of a bottleneck. So those are the same initial population size and current population size. However, the solid line is assuming an immediate one generation. The bottleneck occurs in one generation from the initial to the current size, whereas the dotted line is showing a more gradual decrease in population size, which one might argue is a lot more realistic for most cases. And so you see that there's much decreased power in that case when we have when we're looking at a more realistic model of population decline. So that wraps up the first postdoc that I did. Here's some general conclusions. We may or may not see a linear function as we increase our sampling effort. Power is definitely going to be situation-specific, study-specific. We might see some cases where you reach a sort of threshold past which there's no point in adding more samples. And then something that people really don't talk about much, but I think is actually one of the more important conclusions of this work, is that some signals are simply too weak to detect with any currently realistic, currently feasible sampling strategy, which means that some studies should probably not go forward with the current technology and current tools that we have. Some studies that are taking place may be doomed to fail. But if we use this approach, we would know that beforehand and choose not to do a given study and put our resources elsewhere. So this is the project I'm going to talk about now is what I'm going to do at NIMBioS. The question is similar. It's how do we design a good sampling strategy, again using the similar simulation-based approach. But the challenge is not sampling for statistical power, but what we want to do is gather enough genetic diversity for a conservation collection. Does anyone know this guy, Nikolai Vavilov? Raise your hand if you do. 2 people. So he was a famous Russian botanist in the early 20th century. He was interested in improving the agricultural production of Russia during a time of famine, crop failures. So he figured that we can go out into the wild populations, the wild relatives of our crop species, collect new traits, new genes from them, and probably improve our agricultural productivity. So he did this in the earliest 20th century. He also established a really large seed bank in what was then called Leningrad. And the story goes that this seed bank, so Leningrad was under siege by the German army during World War II. And the people working at the seed bank, everyone around them in the city is sort of starving because they're under siege for weeks and weeks. The people at the seed bank stayed there and protected the collection. And even though they themselves were starving, they didn't eat this clearly edible collection of seeds and grains and whatnot. And actually, some of these collectors died during this time rather than eat their precious genetic resources. We might be collecting seeds for a couple of different reasons. We might be interested in making a collection similar to these seed banks where we're looking to improve crop varieties. But nowadays there are also other functions of a seed bank, which might be collecting from a rare species as a sort of backup in case the natural populations go extinct. In some cases, we're also collecting from common or rare species in order to establish a seed orchard, which is a place where we make lots and lots and lots of seed in order to reintroduce or restore a habitat. So there are a couple of different reasons to collect seed. And for all of them, the goal is kind of to, I guess the first goal is to keep this sort of captive population going over time. But really, in a lot of cases, we're looking to capture alleles of genes that have adaptive value. However, in very few cases do we know what particular alleles will increase a species' fitness in natural conditions. There's a bit of work that's been done primarily in forest trees. Andrew Eckert and David Neal at UC Davis have they've identified some alleles that are associated with a pretty loose correlation to particular environmental variables, but we rarely know exactly what alleles are needed for a species' adaptations in the future. Furthermore, we don't know actually what the future conditions will be. So in most cases with a collection, we're trying to capture just a large number of alleles, and of course to avoid sampling a lot of relatives, a lot of related individuals that will then be inbred. There are guides for how to do a seed collection now. The principal guidelines were developed in the 1990s, and they rely on about the simplest genetic model you can think of, which is just to picture the species as a big bowl with all of the alleles as sort of different at different frequencies in this bowl, and using just very simple equations, you can calculate the probability of grabbing a given allele given a number of repeated draws from that bowl. It's not a very realistic way of planning a collection, but it's actually very widely used. But I think we can improve upon this very simple way of planning a collection by considering what we know about plant biology and plant genetics. We know that throughout a species range, typically there's some level of population structure due to reduced migration between populations, such that certain alleles might only occur in certain populations. Other alleles might be shared widely throughout the range, but be more frequent in some populations than others. We might see a decline in some allele frequencies as you move from one edge to the other. But population structure is a relatively ubiquitous aspect of plant genetics. And in some species, population structure is much more strong than in other species. We also know that within a population, there's typically limited dispersal, which means that within a population, looking at a population in space, there's typically some clusters of related individuals because the seeds don't disperse very far. So we've got some spatial structure within populations as well as between populations. And I think we can consider both of these things when planning a seed collection. We also have to think about the reality of the collectors themselves. So a lot of, so whoever's doing the collection typically has limited resources and therefore limited time to spend out in the field gathering their samples, limited time and money to spend out in the field. There's also political or agency restrictions. So to use butternut as an example, it occurs across the boundaries of some 15 to 20 states, as well as across the border between the US and Canada. So both state or provincial and national agencies may be limited in how much they can collect across the range. There's also within, we have to admit that there's some within population biases due to things like topography. This is the Shenandoah National Park on the border of Virginia and West Virginia, where we made one of our collections. And most of our samples from this population are from along roadsides or along trails because it's a very big park. It's a very intimidating landscape, and we have limited time and energy. So considering these various realistic aspects of plant biology as well as the realism we have to think about in terms of collector strategy, we're making a couple of decisions. The number of samples, the number of populations, what spatial distribution of populations across the range, and then what spatial distribution of individuals within a population. By spatial distribution, I can illustrate spatial distribution with this simple graphic. So if you think of the range of a species as this black box, each circle being a population, the white ones being ones that we actually took samples from. This is sort of a regular, evenly spaced strategy, maybe a grid strategy. This is spatially biased, where we are only able to collect from one geographic portion of the range for various reasons, which I explained. And then this is sort of an edge focus, which we might use if we're interested in capturing rare traits that might occur on sort of the margins of a species range. So those are just a couple of examples of spatial strategies. You can probably think of others, such as a transect or random sampling. So what I propose to do is similar to what I did in my first postdoc, is this approach: simulate populations that represent the real world as it is, test a number of different spatial sampling strategies, as well as a number of different numbers of populations and individuals. But again, just like in that in the first part of the talk, we don't know all of the parameters of some of these important processes that have to go into this model. We don't necessarily know. We probably know a little bit about population size and maybe a little bit about dispersal, but we don't always know what the sort of demographic history of a population is. And these parameters affect how strong that spatial structure is, and therefore the effectiveness of our sampling. So within these simulations, it would be wise, I think, to simulate a couple of different patterns, underlying patterns of the genetic structure, so maybe some clinal variation from one edge to the other, to environmental gradients, habitat suitability, center marginal patterns, or recent declines or fragmentation caused by various things. We want to choose a range of realistic parameters for our simulations. These realistic parameters might represent particular species. We might want to do a species like the chestnut, which has very wind-pollinated animal dispersed seeds, so pretty good dispersal, versus Eryngium alpinum, that other species I showed you at the beginning, which is gravity dispersed and insect pollinated, so much more limited dispersal. That's where we can get some realistic parameters from. And then for our simulations, we also have to choose what genetic markers or traits we're interested in. So in most cases, studies are sort of interested, like I said, in the number of alleles. But with these simulations, we could simulate simple single gene markers that contribute to fitness, or even complex genetic architecture as well. The last thing we need to do before doing these simulations is decide on what we mean by success of a study. So it might be the proportion of the total number of alleles, some measure of genetic distance between those alleles, which might be disconnected from that, the actual trait of interest, how much of that range of trait values do we capture, and then how related is our seed collection. So now that we've thought a little bit about what needs to go into these simulations, this is the workflow that I'm proposing for my project to simulate some of the major processes of interest, range expansion, etc., to create genetic data sets that might represent the natural populations as they are, which we can check the realism in some cases by comparing to empirical data. Then, on each of these simulated data sets, which represent sort of the species as a whole, take various numbers of samples with various spatial distributions and quantify the performance of each sampling strategy by whatever means we decided on the last slide, whatever performance measure we're using. So like the first part of the talk, what we're going to end up with is the relative performance of a given sampling strategy in a given situation for a given species. So what do we do with that? Well, I think there's a number of scientific questions of interest, as well as a number of practical questions of interest. A simple null hypothesis would be that under a given genetic pattern or process, so this is a Kleinel variation in allele frequencies, so the null hypothesis might be that each of these sampling strategies, random, grid, corner, or edge, that they all perform equally well, which a priori we might propose as our alternative hypothesis that this one, the corner strategy, might perform less well than the grid strategy because it's only getting this portion of that Klein. So that's a null hypothesis we can test. Another one is that, so if we take a given spatial strategy, in this case the edge focus, and apply it to each of these different underlying patterns, the null hypothesis is that this given strategy performs equally well across all of those patterns. A couple of other null hypotheses we might be interested in is do the actual parameter values of population size or migration rates, how much do they affect the performance of a spatial strategy? And then the last one might be how much does it matter what type of genetic diversity that we're interested in, maybe neutral or adaptive traits, which might be expected to have different frequencies and then thus different performances. We've also got some practical questions of interest to the people in the field. Practically, simple questions that a person working at an agency might ask is there a sort of general all-purpose strategy that can be applied in across most situations? So can we reach one or a small number of general successful study sampling strategies? And then at what point can we stop collecting seeds? So at what point are we really not gaining much more diversity by collecting more and more seeds? Those are the sort of practical questions. There's a couple of important challenges to this plan that I've outlined. I think there's 4 major challenges. One is to make the simulations realistic. It will be especially challenging to think about what are the appropriate genetic architectures for various adaptive traits, because we don't know a lot about that right now. Another challenge is to include some aspects of the cost of each of these collections. So we have a couple of places that costs come in. We have a cost per sample. That's sort of the amount of time we collect for a given sample. A cost per population, which is more of a transportation cost. You also have fixed costs, that is the cost of the maintaining the collection itself through time. So we can maybe incorporate these given costs to predict how much we're actually spending per new allele or per new sample that we add to the collection. We might, further thinking about cost, we might want to optimize a collection when we have a given limit to the collection size. Say we have a seed bank that can only hold a maximum of maybe 1,000 samples per species. Given that limit, which populations are most important, how many samples per those populations, and should maybe we be sampling more from the populations on the edge or more from the populations in the center, that kind of question. And the last challenge, so these 3 are methodological challenges. The last challenge is a people challenge, and that is getting the results actually applied in the real world, which takes contacts with people working in agencies like the Bureau of Land Management, the Forest Service, various botanical gardens that are doing these collections. So that contact and that communication, but also convincing them that the simulations themselves are reliable, which can be very difficult for some people. The other aspect of this challenge is that many, probably most organizations have years and maybe decades of reliance on an already established sampling strategy. So for example, the BLM, their seed collections, they've been using the same protocol for a given number of years, and convincing people to change that protocol is difficult, even if you can prove to them that another way might be better. So that said, I've given you the outline of what I plan to do at NIMBioS. I'm going to show you about 3 or 4 minutes of one section of results, and then about 3 minutes of another section of results, and then we'll wrap up. So in my proposal, and since being accepted, I've worked on this simple model of what you might call ecoregions, or what population geneticists call hierarchical population structure. That's where we have a given range. Each white box is a population. The populations within a region are better connected, higher migration rates than those between regions. That's what we call hierarchical structure. So given this simple model, for which we have some evidence in empirical populations, we can test 2 very simple strategies, that is, collecting all the populations in one region, or collecting one population from each region. You see in this simple example, it's the same number of populations, but with different spatial distributions. The result I have so far, the spread strategy, which is this one, always performs better, but the relative advantage of that strategy over the targeted strategy depends on the parameters of the simulation, so the species, and it depends on what type of allele that we're interested in. So let me show you these results. It's a bit of a complicated slide, but let's focus on the first column. Each column is a different category of alleles. So we might categorize alleles by where they occur and what frequency they're at. So let's first just talk about globally rare alleles, which are throughout the whole range, those that occur at a frequency of lower than maybe 0.1 or 0.05. The black line is a strategy where we sample actually all the populations. The dotted line is where we use the spread strategy, and the dashed line is where we use a targeted strategy. Again, these two have the same number of populations, just different spatial distributions. And you can see it's pretty dramatic how much better the spread strategy performs for this species that has relatively low connectivity. So that's relatively low migration rates and therefore relatively high population structure. You see that as we look at species with moderate or high connectivity, the relative advantage of this one strategy over the other decreases. The pattern is also there to some extent for each of the other categories that I show here, those that are restricted to one region or one population. You can see the relative advantage of the spread strategy over the targeted strategy might decrease as you increase connectivity for that species. So these results are what we might expect. For a long time, people have said it's best to sample as widely as possible. So to spread your samples out, if possible, throughout the range, but no one has really quantified the relative advantage of that strategy for realistic parameters. I'll skip that one and get to the, so this first thing I was talking about was our strategy for which populations, how many populations. This is within a population. I was working with Alan Strand, who was visiting NIMBioS back in the fall. We did some simulations of individual-based simulations in which you have these individual trees distributing pollen, being pollinated, distributing seeds. You have realistic age or stage transitions. You've got some variation. You can see pretty realistic variation across the population, some clumping, which is pretty typical for plant species. So he worked with me to create these simulations, and then I've been working over the past couple weeks on trying out various sampling strategies, one of which you see here. This is a grid sampling. You notice that the chosen individuals are not exactly on the grid point, partly because of a little variation in where they occur, and partly because maybe you have a maternal tree here, but it didn't bear seeds that season. So there's also some variation in how many seeds each mother tree creates each season in these simulations. So we might choose one not actually on that grid point. So I'm going to show you the relative performance of each of these spatial sampling strategies within a population as measured by the proportion of alleles caught. And I just got these results over the weekend, so they're not very well developed yet. But the first thing that pops out at you is that this strategy, which is that restricted to maybe one corner of the population due to access or other reasons, laziness, performs the worst. Whereas the grid sampling and the random sampling perform the best. And then you have a transect, which is just one line going right through the population. That performs, shows intermediate performance. In this case, it actually shows basically equivalent performance. Again, I haven't put error bars on here, but we can measure the statistical, whether these strategies are statistically different from each other. I just haven't done that with the results yet. This is for sampling from 4 seed trees and then 9 seed trees, and then this is more seeds per tree. You see diminishing returns as you sample more seeds per tree. I have a lot of parameters to play with for these simulations. I won't go through them all, but I'll have to think about different dispersal characteristics, different molecular markers, annual or perennial species, and then different processes that might have affected our population of interest. To conclude, my work in my first and second postdoc have shown that we can use simulations to better plan sampling strategies for detecting genetic effects and also for collections for breeding or conservation. The main conclusions are that the sampling strategy differs very much for each species or study we're interested in, and that spatial arrangement might be more important for species that are not well connected. I've been fortunate enough to work with a number of different institutions and partners at different institutions who I'd like to recognize. A number of people who were important during both my PhD and postdocs, people who've given me money to travel or to do work. And these are all the people who helped with the collections of butternut, a lot of private landowners and a lot of partnerships with botanic gardens and other people. And now maybe we'll have a few questions.